Good. Well, good morning, everybody. It's great to be back at the MESA meeting. Australia was the most locked down country in the world during the COVID pandemic, so getting out and about again is very exciting for us. My name is Macdonald. I'm the CEO at Cynata Therapeutics. We are an Australian stock exchange listed mesenchymal stem cell company developing a unique platform for the production of therapeutic mesenchymal stem cells. A few words here, just our safe harbour statement. As we are a publicly traded company, please refer to our website and to our announcements on the Australian Stock Exchange platform for further information about the company. A snapshot overview of our company and our activities. We, as I mentioned, are developing a unique and proprietary platform utilising iPSCs for the manufacture of allogeneic mesenchymal stem cells or mesenchymal stromal cells. We are the leading company worldwide in the development of iPSC-derived cell therapies of any kind, and as you will see during the presentation, we have multiple clinical trials underway. We're the first to conduct a clinical trial using an iPSC-derived cell therapy. So the excitement around the application of iPSCs is very clearly manifested in Cynata's portfolio. We've developed a strong safety and efficacy portfolio, both clinically and preclinically, in a range of different therapeutic targets. We have a strategic partnership with Fujifilm, a powerhouse in regenerative medicine and manufacturing. We have multiple clinical trials underway and recently received clearance for an IND here in the United States. So again, a major milestone for the iPSC space and obviously an enormous catalyst for the company. Large addressable markets served by our potential applications and of course significant valuation upside as a publicly traded company if we are successful in our ongoing programs. There's been an enormous literature over the past couple of decades about the functionality of MSCs and why they work. And this slide obviously is not intended to be read through during this presentation, but it's just to give you an idea and a summary that MSCs now are well recognised as the sensor and switcher of the immune system, affecting most of the functional elements of our response to invading organisms, to repair and regeneration. So, a lot of questions are asked about a mechanism of action of MSCs, and I think now we have a very clear picture of how this very interesting functional cell type has profound effects upon the immune system. And this has led to an enormous amount of research and development as a range of targets are being investigated clinically around the world. Around 1,200 clinical trials have been initiated using MSCs in the past decade, and it's important to remind the audience that there are already approved MSC-based products on the market, both in Japan and in Europe. This widespread interest ultimately in the efficacy and safety of MSCs now has translated to an enormous amount of interest in how the product is manufactured, because at the end of the day, even if you've got a very safe and very effective product, if it can't be manufactured economically, consistently, and at scale, then you don't have a product. And Cynata's platform, utilising iPSC, addresses that very significant need. And quite simply, our process is illustrated here in this cartoon. On the top panel, you'll see the conventional process of production of MSCs, which, as most of you will be aware, relies on donor-derived tissues such as bone marrow or adipose tissue or placenta or other sources. Multiple donors, the MSCs are isolated from the gamish of cells in the particular tissue. They're massively expanded in culture, and then that results in the finished product. There are multiple flaws in that strategy which are addressed by an iPSC approach, which is our strategy in the lower panel, where we utilise a single donor to derive an iPSC, which then forms the basis of our manufacturing starting material forever, thus ensuring that we have a highly consistent product. We can manufacture the product at scale because the expansion is at the level of the iPSC, thus avoiding the very, very clear problem of lack of potency and senescence that occurs when MSCs are expanded in culture. and it also assures that we have a very efficient product. And some of our clinical data has shown that our cells are much more potent than their equivalent derived from, for example, bone marrow. But most importantly, it's all well and good for a company such as us to say, hey, we've got the best platform around for manufacturing a cell therapy product, but it's ultimately the arbiter, the regulatory agencies, who have the final decision. And it was a very, very important data point from the FDA a couple of years ago when they issued a complete response letter in response to an application for a BLA for an MSC product. And the FDA had a number of issues with that BLA, as some of you may be aware, but their commentary was incredibly telling for the industry. And that is, you guys have got a problem if you're relying on multiple donors and you haven't demonstrated to us that you've got consistency from donor to donor, because fundamentally, we're all different. And so please show us that you've got a product that is consistent from donor to donor and batch to batch. And that wasn't demonstrated in that BLA to the FDA's satisfaction. On the other hand, Cynata's technology, because we use the same starting material every time, we can assure that, at least as far as donor differences are concerned, that's not a problem for us. Of course, we still have to show that we've got consistency. But that was a very, very illustrative commentary from the FDA in response to an application here for an MSC product. Let's look now at our clinical data. The most exciting data we have so far was from a phase 1 clinical trial we conducted in acute GVHD for our first product, CYP001. 7 sites in the United Kingdom and Australia, and these were patients who really have very few options. Acute GVHD that fails to respond to corticosteroids is a very, very poor outlook. The study data, in summary here, we assessed the patients at the primary readout at day 100, and we saw a complete response in more than half of the patients, an overall response in nearly all of the patients, and a very good survival rate, again, in nearly all of the patients. Taking this out to 2 years, we saw a very durable response in these patients, where nearly 2/3 were still alive after 2 years. And those of you who know GVHD will understand that the outlook for those patients at 2 years is normally very grim, and typically around 17 to 40% or less are still alive. So this was a great result. It was a great trial for the sector for iPSC-derived cell therapies. It resulted in a front cover article in Nature Medicine magazine as being the first clinical trial utilising an iPSC-derived cell therapy product. That great data has now allowed us to move forward into multiple clinical targets. We have a phase 3 clinical trial underway in Australia in osteoarthritis, a trial underway in diabetic foot ulcers, and under a cleared IND here in the United States, we expect to get a phase 2 clinical trial in acute GVHD underway sometime in the coming months. We're just in the process of selecting and validating clinical study centres, both in the US and Europe, as well as in Australia at the moment. We have additional targets that we will pursue as finances and resources permit in areas such as respiratory distress, critical limb ischemia, IPF, and renal transplantation, all areas which are substantiated by a solid body of preclinical data, not only from our cells, but other MSCs as well. Turning now to a brief description of our clinical programs. As I mentioned, we anticipate commencing a phase 2 trial in acute GVHD under a cleared IND in the US and other sites. These patients, as I mentioned before, have very few options once they've failed corticosteroids. The largest clinical study probably ever with an MSC product for osteoarthritis is now underway in Australia. This is a double-blind, placebo-controlled, randomised, 440-patient study. So it is a landmark, gold standard clinical trial in this very challenging condition. Osteoarthritis is a progressive condition for which there is no cure. The only treatment is analgesics and lubricant-type products that are injected into the synovium, but ultimately the patients progress, and assuming they still lead an active life, usually that results in a hip replacement, knee replacement, some form of joint, artificial joint inserted, and then the patient resumes function. But of course, wouldn't it be great if we had a product that we could inject into the patient that avoided surgery, that ultimately resulted in a cessation of the degradation of the joint, or better still, we got an improvement in the joint function through regeneration of the cartilage tissue. And that's the hypothesis that we're working on. So endpoints include, obviously, mobility and pain, but also an MRI assessment of the joint to show whether or not the MSCs are having that disease-modifying effect. If that is the case, of course, this will be a major breakthrough for a condition which enjoys a very, very large patient population globally. Diabetic foot ulcers are a very challenging condition, the incidence of which continues to rise as diabetes increases around the world, particularly in countries that are not as au fait with enjoying a healthy lifestyle and diet. Challenging condition for which little has changed in the last 100 years. This trial is now well underway in Australia. It commenced patient recruitment earlier this year, and it involves a topical application of our cell therapy product. Some few words on preclinical data. Often we're asked, oh, you know, your MSCs, that's a really cool way of manufacturing them, but are they better? Well, that, of course, is a very subjective question, but we have conducted a number of studies where we've investigated the potency of our cells in well-accepted preclinical models compared with bone marrow MSCs or MSCs derived from other sources. One of those was in a rodent model of myocardial infarction, where we were looking at the enhancement of recovery of the infarcted area of the heart following this model. And there, the comparator was bone marrow-derived MSCs. That's the conventional MSC, if you like. And in nearly all of the indices in this study, we saw an enhanced effect in this model using our MSCs, our iPSC-derived MSCs. Likewise, in a model of diabetic wounds, again, the comparators were, for example, bone marrow MSCs and MSCs from sources like dental pulp and bone chips. And there, again, as you'll see in the dark blue bar, our Cymerus MSCs, our iPSC-derived MSCs, were more effective. So whilst this is only preclinical data and would need to be confirmed in head-to-head studies in the clinic, we're very confident that this data will translate to improved potency, and we certainly have the improved consistency. One index of that was our GVHD study, where the dose required to produce the clinical effect that I explained before in the earlier slide was 2 doses of 100 million or 200 million cells per kilogram. And that compared very favourably to the typically 8 to 12 similar doses that are required for bone marrow-derived MSCs in the same disease. So that, of course, represents a huge competitive advantage. It's a major cost advantage and a cost per treatment advantage, which, if it's borne out, will underscore the high potency of our cells, and that potency, of course, is preserved through the manufacturing process. We have a strategic partnership with Fujifilm, who, as I mentioned before, are a powerhouse in regenerative medicine, particularly in the manufacture of cell and gene therapy products. We derived our iPS cell line from the then Cellular Dynamics International in Madison, Wisconsin, which is now a Fujifilm company, FCDI, and we're utilising FCDI's resources for manufacturing scale-up and for eventually commercial manufacture. Looking now at near-term catalysts for the company, clearly getting good clinical data is the major value driver for biotech companies, and we've heard that time and time again during the past couple of days. Good, solid clinical data with well-designed clinical trials. So completing our DFU and our DFU trial is certainly one of the key drivers of value. We just had a DSMB review this week, so this slide's a fraction out of date, but the DSMB provided clearance for that trial to continue. We expect to complete recruitment in our osteoarthritis trial in association with the University of Sydney, and then obviously announcing our DFU results during the course of next year will be a major catalyst for us. We have a very clear strategy on partnering our technology. We are a small Australian company, and accessing global markets through partners is an essential part of our business strategy. So we have a very vibrant business-to-business engagement program, seeking partnerships geographically, therapeutically, by platform. Essentially, all options are open and available. Finally, just a quick view of the company's makeup. We have three major shareholders: Fidelity International, Bioscience Managers, an Australian UK venture fund, and Fujifilm, our strategic partner. Together, they're about 25% of our share register. The company is valued at about $31 million, a little more in the last couple of days following our DSMB review. And we have about $16 million in cash, which doesn't sound a lot for a US biotech company, but Aussie biotechs run very lean and mean, and our burn provides a significant runway well out into the coming year and beyond. So I'd like to thank you all for your attention. If there's any interest in discussing our iPSC-derived MSC platform, please refer to this slide where you can find our website and also my email address. I'm sorry I can't stick around for questions. I've got a one-on-one meeting coming up right now, but thank you very much for your kind attention, and thank you to the ARM for the opportunity to speak.